I am Dr. K.AMUDHA from Biochemistry department. In today's topic, we can see serum marker enzymes in liver disease. The major enzyme which is involved in liver disease is serum transaminase, that is AST and ALT, and the other enzyme is serum alkaline phosphatase, that is ALP. And not only that is increased, there are some other enzymes that is also increased. These enzymes are serum 5-nucleotidase, serum leucine aminopeptidase, serum isocitrate dehydrogenase, serum hydroxyl butyrate dehydrogenase. The next enzyme is serum aldolase. Then it is serum choline esterase. Then serum ornithine transcarbamyl transferase, serum amylase, serum sorbitol dehydrogenase, and serum gamma glutamyl transpeptidase. So going on for the first enzyme, it is transaminases. So transaminases are the enzyme which transfer amino group from one amino acid to keto acid for the formation of new amino acid and keto acids. And this is done by the enzyme transaminases, and it needs a coenzyme, and that is called as a pyridoxal phosphate. This AST is present in cytosol and also in mitochondria, and ALT, it is located in cytosol of the liver. And this is important in assessing and monitoring the degree of liver cell inflammation and also necrosis. The highest activity of the transaminases, that is ALT, are found in hepatocytes and also in muscle cells. Elevations in serum ALT are considered to be relatively specific for liver disease. During viral hepatitis, there is a 100 to 1000 times increase in both ALT and AST, but ALT level is increased more than that of AST. Now we'll see about the transaminases enzyme, that is aspartate aminotransferase, that is AST. This aspartate aminotransferase, it transfers amino group from aspartate to alpha ketoglutarate and forms glutamate and oxaloacetate. And this enzyme, it is also called as serum glutamate oxaloacetate transaminase, that is SGOT. And this needs pyridoxal phosphate as a coenzyme. The normal serum level of AST is 8 to 40 units per liter. So when you want to see the distribution of this enzyme, it is distributed in liver, heart, skeletal muscle, kidney, and also in erythrocytes. The marked rise of this enzyme is seen in circulatory failure with shock and hypoxia, and during myocardial infarction, and also during acute viral or toxic hepatitis. When it comes for the moderate increase, the moderate increase of this enzyme is seen in cirrhosis, cholestatic jaundice, malignant filtration of the liver, and also during skeletal muscle disease. Transaminase enzyme is alkaline aminotransferase, that is ALT. Here, the L-alanine combines with alpha ketoglutarate and it is converted to pyruvate and L-glutamate, and this is done by the enzyme alanine aminotransferase. And this enzyme, it is also called as glutamate pyruvate transaminase, that is SGPT, and pyridoxal phosphate acts as a coenzyme. Here, the normal level of alkaline phosphatase is 5 to 30 units per liter. Very high values, that is 100 to 1000 units per liter, are seen in acute hepatitis, either toxic or viral in origin. Both ALT and AST are increased in liver disease. When it comes for the distribution, high concentration of this enzyme is seen in liver, whereas lesser extent is seen in skeletal muscle, kidney and also in heart. The marked rise of this enzyme is seen in circulatory failure with shock and hypoxia, and also during viral and toxic hepatitis. The moderate increase of this enzyme is seen in cirrhosis during infection, liver congestion secondary to congestive cardiac failure, and during cholestatic jaundice. Next, we'll see the enzyme alkaline phosphatase, that is ALP. This enzyme, it is a non-specific enzyme which hydrolyzes aliphatic, aromatic or heterocyclic compounds. The pH optimum for this enzyme reaction is between 9 and 10. The normal level of alkaline phosphatase is 40 to 125 units per liter. In children, the upper level of normal value may be more because of the increased osteoblastic activity. So when I come for the distribution, it is produced by osteoblasts of the bone, that is bone cells, and localized in the cell membrane, and it is also present in lactating mammary gland and in placental. When you see the moderate increase, 2 to 3 times, that is increase in alkaline phosphatase level, is seen in hepatic disease such as hepatitis, alcoholic hepatosis or hepatocellular carcinoma. Very high levels of ALP, that is 10 to 12 times of the upper limit, may be noticed in extrahepatic obstruction or cholestasis. So drastically high levels of ALP, that is around 10 to 25 times of the upper limit, are seen in bone disease where osteoblastic activity is enhanced, such as Paget's disease, rickets, osteomalacia, osteoblastoma, metastatic carcinoma of the bone, and during hyperparathyroidism. A major marker enzyme which is released during liver damage is SGOT, SGPT and alkaline phosphatase. And there are also other enzymes which is increased during liver damage. So we'll see all the other enzymes one by one. The first enzyme is gamma glutamyl transferase, and shortly we can say this as GGT. This is the enzyme involved in amino acid transport across membrane, and this is used in the synthesis of glutathione, that is a non-enzymic antioxidants. This GGT exists in isoenzymes form, and there are 11 isoenzymes present in this. So going on for the distribution, it is seen in liver, kidney, pancreas, intestinal cells and also in prostate glands. The level of the enzyme is increased in the tissue kidney and it is low in the heart tissue. When it comes to the normal value for this enzyme is 6 to 45 units per liter in male and 5 to 30 units per liter in female. This GGT is marked increase during alcoholism and the moderate increase is seen during liver disorder. The GGT level is highly elevated during alcoholism and also during obstructive jaundice. The isoenzyme GGT2 is positive for 90% of hepatocellular carcinoma. Increase in GGT level is generally proportional to the amount of alcohol intake. Next enzyme is nucleotide phosphatase, that is NTP. And this is also known as 5-nucleotidase. This enzyme hydrolyzes 5-nucleotide to nucleoside at an optimum pH of 7.5. This is a marker enzyme for plasma membranes. AMP is used as a substrate which is hydrolyzed to adenosine and inorganic phosphorus. When you come for the normal level of NTP, the level in serum is 2 to 10 units per liter. And this enzyme is moderately increased in hepatitis. And this is highly elevated in biliary obstruction. We'll next go for the next enzyme. It is serum choline esterase. This enzyme is present in the nerve endings and also in the RBCs. The normal value for serum choline esterase is 2.17 to 5.17 IU per ml. The concentration of this enzyme is increased during recovery from the liver damage, that is actively growing hepatocytes, and the concentration is decreased during hepatic parenchymal disease. Other enzyme which is increased during liver damage is serum amylase. The liver is a major, if not the only source of amylase found in the serum under normal physiological conditions. The normal serum level is 50 to 120 units per liter. And the studies have shown low serum amylase levels in liver disease like acute infectious hepatitis. Next, coming for the next enzyme, it is serum aldolase and phosphohexosisomerase. Both these enzymes are markedly increased in serum of patients with acute hepatitis, and no increase is found in cirrhosis, latent hepatitis or biliary obstruction. Going on for the next enzyme, it is serum leucine aminopeptidase. So shortly we can say this as LAP. This is a proteolytic enzyme which splits off N-terminal residues from certain L-peptides and amides having a free NH2 group, especially when the N-terminal residue is leucine or related amino acids. So இந்த serum leucine aminopeptidase என்றது ஒரு proteolytic enzyme, protein cleave பண்ற enzyme. So இது என்ன பண்ணும் அப்படின்னா ஒரு L peptide இருக்கிற N-terminal amino acids இருக்கிற leucine or related amino acids cleave பண்ணும். The normal range of this enzyme is between 15 to 56 milli-IU. Mild to moderate increase is seen in viral hepatitis, and this ranges from 30 to 130 milli-IU. Increase is also seen in cirrhosis of the liver, but rise is less. Marked rise have been seen in liver cell carcinoma. Next, we'll see the enzyme serum hydroxybutyrate dehydrogenase. Shortly we can say this as SHBD. This is an enzyme acting on hydroxybutyric acid identified in the serum and studied as a diagnostic aid in liver disease. So the normal level of this enzyme in serum is between 56 to 125 IU per liter. This enzyme, it is elevated in acute viral hepatitis, and it is also elevated in myocardial infarction. So in this we are supposed to see the ratio of LDH by serum hydroxybutyrate dehydrogenase. This is to differentiate the liver disease and acute myocardial infarction. So the ratio of LDH by serum hydroxybutyrate dehydrogenase has been found more useful. The normal ratio should be 1.18 to 1.60. So less than 1.18 is observed during myocardial infarction. Greater than 1.60 is observed in liver disease. In infectious hepatitis, the ratio is frequently more than 2. And in chronic hepatitis and also during obstructive jaundice, the ratio ranged from 1.6 to 2. The next other enzyme is serum sorbitol dehydrogenase, that is STH. This enzyme catalyzes the conversion of sorbitol plus NAD by the enzyme sorbitol dehydrogenase. It is converted to fructose and NADH. The normal values of STH in serum is less than 0.2 MIU. There is a striking elevation seen in acute viral hepatitis and also during carbon tetrachloride poisoning up to 17 MIU. In chronic hepatitis and also in obstructive jaundice, the serum levels of sorbitol dehydrogenase are normal or they are slightly elevated. Coming for the next enzyme, it is serum isocytate dehydrogenase, that is ICD. This is a specific enzyme which is found in the liver only. The normal range of this enzyme is 0.9 to 4 IU per liter. In liver disease, a marked increase in infectious hepatitis and malignancy or after taking drugs. There is a large increase seen in infectious hepatitis. The serum activity almost returns to normal by the 3rd week after the onset of jaundice. In most cases of cirrhosis of the liver, the serum enzyme activity is either normal or slightly raised. So coming for the last enzyme, it is serum ornithine carbamyl transferase, that is OCT. Ornithine combines with carbamyl phosphate by the enzyme ornithine carbamyl transferase. It is converted to citrulline and phosphate. This enzyme, it is exclusively found in liver and no activity is seen in other tissues. The serum enzyme activity is normal. Healthy adults are usually very low and this ranges from 8 to 20 MIU. In liver disease, there is a markedly elevated levels of this enzyme, that is 10 to 200 fold in patients with acute viral hepatitis. Relatively, slight elevations occurs in obstructive jaundice and cirrhosis of the liver and also during metastatic carcinoma. In this topic, we have seen what is clinical enzymology and what are all the enzymes are increased during liver damage, the major enzymes and the other enzymes which are released during the liver damage. Thank you. Thank you. Thank you.